Hello, and thank you for joining us for the 49th Annual AAKP National Patient Meeting. We hope you've been enjoying the sessions. My name is Jonathan Haydak, and I am a former dialysis patient and current kidney transplant recipient from New York. I have the honor of being a newly elected member of the AAKP National Board of Directors, and I have been an AAKP Ambassador since 2018. Being an AAKP Ambassador has provided me the opportunity to advocate for patients and offer their perspectives to lawmakers, industry leaders, and various research consortia. As a PhD student researching kidney disease, my scientific expertise helps ensure that patient voices are central to developing effective and patient-centered policies and research. As a proud member of AAKP, I firmly believe that every kidney patient's voice is paramount in determining their care plan. It's essential that we cultivate an environment that encourages kidney disease research and innovation, driving continuous advancements in care, improved treatments, and solutions for unmet patient needs. Once these innovations receive FDA approval, it is equally important that all patients have access to and the right to choose the best treatment for them in consultation with their chosen healthcare team. This ensures every patient can live their best life and achieve their aspirations. With that, it's my honor to introduce our next session, titled Advocacy and Engagement Training, From Concept to Consumer, Understanding the Product Development Lifecycle. We are excited to host this session as part of AAKP's ongoing training efforts to provide you with the practical skills and knowledge to be effective in a variety of efforts and activities. Since 2022, AAKP has developed a series of training sessions, such as being effective with reporters, how to talk to elected officials and staff, maximizing your social media platforms for advocacy purposes, how to be an effective participant in clinical trials, and how to effectively collaborate with researchers and principal investigators in research study opportunities. These are just a few of the 20 on-demand skills-building sessions AAKP makes available at no charge on our YouTube channel. Today's session, From Concept to Consumer, Understanding the Product Development Lifecycle, will take us through the journey of how a brilliant idea turns into a product that truly impacts patients' lives, and that is no easy feat. The process involves understanding patient needs, conducting thorough research and development, navigating regulations, and strategically entering the market. This session aims to provide insights into each step of this process and highlight the collaborative efforts required to bring innovations to consumers. At AAKP, we've always championed patient-driven innovation. We advocate for patient involvement in product development and early-stage research, as well as for the fostering of partnerships between patients, clinicians, and industry leaders. Through initiatives like our Center for Patient Research and Education, we ensure that the voices of kidney patients are heard and that their experiences shape the product and therapies designed for them. Today, we are honored to welcome a valued friend and distinguished guest, Dr. Tejas Desai. Dr. Desai is a nephrologist in Charlotte, North Carolina, and a former member of the Board of Directors for AAKP. He began his career in academia as a training program co-director for the Internal Medicine Residency Program and then Nephrology Fellowship Director at East Carolina University Brody School of Medicine. Following his career in academia, he joined the Department of Veterans Affairs and was privileged to serve our veteran CKD and ESRD populations. He joined CSL Vifor as Senior Medical Director to continue contributing positively towards the health of kidney patients. Dr. Desai, I turn the presentation over to you.
Thank you, Jonathan, for the warm introduction, and thank you to the AAKP organizers and board of directors for the opportunity to present to you today at the 49th Annual AAKP meeting. My name is Tejas Desai. It's a pleasure to be here. We'll be talking about from concept to consumer, understanding the product development life cycle of a medication. My name is Tejas Desai, and I work for CSL Vifor, and we'll be speaking about one of the therapeutic agents that CSL Vifor has brought to patients in the nephrology community from a concept all the way to the patient or the consumer, and that is patiromer. Just so you know, The views and opinions expressed here are of my own and not necessarily of CSL Vifor. Just a little bit of background of who I am, and you can certainly revisit this at a later date. But I do work for CSL Vifor. I've been with the firm for 2.5 years now. It's been a pleasure working for this company as we are committed to the nephrology community and patients who are afflicted with kidney disease. Prior to my joining CSL Vifor, I had the pleasure of working with veterans in the United States Department of Veterans Affairs in Charlotte, North Carolina, which is where I am based. Because we'll be talking about patiromer and using its entire journey as an exemplar of how we take something from a concept and bring it to the consumer, to the patient, I do want you to be aware of the indication and safety information about this medication. Throughout this presentation, you may have questions about the journey of a medication from its concept to the consumer, and I would certainly entertain those questions. If you have any questions about patiromer itself and its indications and how it's used, those questions are best left for you and your physician to discuss. So we've broken down the drug development process, concept to consumer, into 5 large buckets, and you'll see those buckets here on the screen. We'll start with discovery and development, and we'll end with our post-marketing efforts. Just so you know, as an employee of CSL Vifor, I primarily operate in the last bucket, in the post-market efforts, but I also have an opportunity to work with discovery and development, and then right in the middle where we see clinical research. We will go through all 5 of these buckets in this presentation. So let's talk about discovery and development, and specifically looking at potassium binders, which is what patiromer is, and its treatment for hyperkalemia, which is the elevated levels of potassium in one's blood. One of the things you have to do in your discovery development process is to understand the disease state, to see how much of a prevalence this disease occurs and whether it actually burdens the patient, and if so, how does that burden manifest? So what we notice is what you see here on the top right, the blue arrow and the red box, that there is an increased risk of hyperkalemia or elevated potassium in the bloodstream when 3 common things happen. 1, if you're afflicted with chronic kidney disease, and that's a common cause of having elevated potassium levels. 2, if you're using a very common and beneficial class of medications called renin-angiotensin-aldosterone system inhibitors, or RAS inhibitors. The most common subclasses of that class are ACE inhibitors or ARB medications. And then 3, if you are exposed to high potassium food. And as we know, a lot of heart-healthy medications have a lot of potassium. And so if you have any one of those 3 or a combination, then your prevalence of having hyperkalemia increases. So there are risk factors for why this disease entity would occur. Now, what's the burden of having high potassium levels? And as part of the discovery process, we learned, of course, that you can have an irregular heartbeat if your potassium levels are elevated, some form of an arrhythmia that could either lead to or reveal a pre-existing heart condition. And some of times, in unfortunate times, if these conditions are severe, it can lead to a possible hospitalization. So there is a risk for having hyperkalemia. That risk is predominant in the kidney community in patients who are afflicted with kidney disease, and there's a burden that comes with having that event happen. In the discovery, we asked ourselves, okay, if hyperkalemia does occur and it is burdensome to the patient, what are the treatment options? And at the time that we did this discovery, there were really 2 main treatment options. One was to reduce the potassium in your diet. We'll talk about that some more in the next slide. And the other one is to use a medication that was in existence at the time to treat hyperkalemia, to eliminate that excess potassium in one's diet. So let's take a look at both of these. So we will take a look at the medication burden first. So we know that in 1958, a medicine called K-Xlate was approved for use to treat hyperkalemia. And this is a bit of the life cycle of that medication. So it was initially published in the late 40s and 50s, then in the early 50s, and then in the late 50s, K-Xlate was given approval to treat hyperkalemia. And then something very interesting happens, and it's in 1962, where the FDA undergoes a modernization and new rules and regulations come into place for any medicines that are going to be going to receive its approval from that point forward. And what's interesting is the word grandfathered. And so K-Xlate was grandfathered into the marketplace. In other words, it did not have to be restudied with these new parameters in place. And anytime there's a regulatory change, whether there's less of a burden or more of a burden, less regulations or more regulations placed on the research and scientific community, it opens the door for new development. And so that grandfather designation means that there are certain questions that KX-Late would not be able to answer because it didn't have to meet that requirement any further. So if we were going to design something new and different, we have to have some attributes of this new medicine that are different than the existing medicine. And so now we're into the development phase. What kinds of attributes do we want to develop in our new medication that would separate it from the old medication and make it presumably better? And so one of those attributes is the surface morphology of the medicine itself. KX-Late is shown on the left. Those are microscopic images of what a KX-Late bead looks like. And patiromer is shown on the right. That's a schematic. What you'll notice right away is that KX-Late has jagged edges. It does not have a uniform morphology. It is asymmetric. And on the right, patiromer was intentionally designed to have symmetry, to be a sphere. Now that spherical morphology, we presumed, would give it certain attributes that were missing in KX-Late. Number one, it would be more palatable and easy for a patient to ingest than something that was jagged. Number two, because it's spherical, it would more likely pass through the GI tract smoothly rather than a jagged molecule. And number three, the presumption was that a spherical morphology would allow more regular absorption or binding of potassium, more uniform sequestration of potassium than something that is jagged and not uniform and asymmetric. So that's one of the attributes that we wanted to have in the new medicine, patiromer, which is part of its development process. The second attribute that we felt should be added to patiromer in its development was the manner in which potassium is bound. And so the potassium binders will bind potassium and have to release something in its place. So it's a swap. And in the case of KX-Late, the swap is usually with sodium. So as KX-Late binds potassium, it must give up the sodium that it has. Patiromer was designed to swap calcium instead of sodium. And there's a reason why that's the case, because we also knew that a lot of foods in the Western diet, while rich in potassium and would result in hyperkalemia, are also rich in sodium. And there are a number of detrimental effects when a patient is exposed to sodium, and in particular CKD patients who are exposed to sodium. At the time, the dietary guidelines for Americans recommended 2,300 milligrams of sodium in a day or less. That has somewhat relaxed now to approximately 3,000 milligrams a day. Again, it depends on which guidelines you're referring to, but for the most part, 3 grams of sodium per day is what's recommended. And there's quite a bit of sodium exposure when you use the previous generation of potassium binders. You'll see that about 1,500 to 6,000 milligrams of sodium can be contained in a 24-hour period of using KX-Late. So we didn't want to use sodium and expose vulnerable patients to a sodium load. So we swapped out the sodium and designed a molecule, engineered it such that it would swap calcium for potassium. All right, so now we're in the second stage. We've done our due diligence, which is usually the first stage, discovery of what's out there in terms of this disease state and what is being done and what those limitations are in the current existing therapy. We're now in the second stage where we'll do preclinical research with what we believe would be a better option for treating hyperkalemia. So a lot of the preclinical research is done in animal models. And we're often trying to determine how the medication works in an organic organism, I should say, like the mouse. And we want to know how it's absorbed, whether it actually does its job of binding potassium. If it does bind potassium, how well does it bind it? And then how does it eliminate potassium? Because in the end, binding potassium is not sufficient. We also have to have it eliminated from the animal such that it can no longer be absorbed or do any damage. So that's what a lot of our preclinical studies focused on. Those don't often get a lot of attention in the scientific community. A lot of medications die at this point because things don't work out, but things worked out for patiromer, and then we moved on to the next stage. So this is the stage that most individuals are familiar with, and it's the clinical research. This is the stage where a lot of the science is far more well-known, far more publicized, and is often presented at academic conferences and meetings such as the one that AAKP has every year. And clinical research is broken down into the three phases: phase 1, phase 2, phase 3. Here we show phase 1 in dark blue, phase 2 in yellow, and phase 3 in orange-red. Now, phase 1 is really focused on safety of the molecule. It's also an opportunity for us to determine what dose would be an optimal dose to study moving forward in the clinical research phase of the journey. And we'll look at our phase 1 study momentarily. Phase 2 is about efficacy. So now that we know what's the best dose or one of the best doses to use in terms of safety and the safety profile, we're going to see how well these different doses work. And then phase 3 is usually considered the comparator phase. So you often compare your molecule at its best dose, whichever dose you feel was the best from phase 1 and phase 2, and compare it against the standard of care, keeping in mind that the standard of care has to be something that is FDA approved. And then we'll talk about what's in the purple and then what's in the green later on. So this is phase 1, where we're looking at both safety and dosing. And what I'm showing you here is the change in potassium, in how much potassium you excrete in your stool on the y-axis. And on the x-axis is the various doses of patiromer that were studied. And you can see placebo in gray, so no patiromer, 0 milligrams, and then different doses of patiromer from 0.8 milligrams all the way up to 16.8 milligrams. This is a dose-dependent response. That's what you're seeing here on the screen. More potassium is excreted as you use more patiromer. And not shown on this slide, but in the paper as well, is that the safety profile is pretty similar across the last 3 doses of patiromer. And so we use this information going into phase 2, which is in the next slide, to determine the efficacy. Can we see a drop in the serum potassium, not just in how much potassium is excreted when we use these doses of patiromer. So here's phase 2, known as the Amethyst study, study number 205. And in this study, we're looking at efficacy. So you administer a particular dose of patiromer. That's done all the way here on the left side of the graph. And you'll see a purple and a green color. Purple represents all the patients who at baseline had moderate hyperkalemia, so potassium level of 5.5 to about 6. And the green represents patients who have mild hyperkalemia, potassium about 5.1 to 5.4. That's the serum potassium on the y-axis and the weeks of the study duration on the x-axis. You give patiromer at the baseline, which is shown all the way to the left, and then you follow the doses, the potassium level over time as patiromer is given on a regular basis across a 52-week period. And what you see here when it comes to efficacy is that, yes, patiromer will drop your potassium into what's considered the normal range, and that's the shaded region between 4.0 and 5.0. And as long as you use patiromer on a regular basis as prescribed, your potassium remains in that range. At the end of the 52-week period, you want to see if efficacy is lost by discontinuing the medication. So when patiromer is discontinued at week 52, you see that in both patient populations, the potassium level starts to rise. And so this is the type of efficacy study that we do in the phase 2 part of the clinical phase of the journey of a medication to determine if there's efficacy in the use of this medication. So now we go to phase 3. And we have about 4 slides here regarding phase 3. So now we're doing a comparison. We've found what we consider to be an optimal dose of the medication, and we're going to compare it to the standard of care. And in the case of hyperkalemia, the standard of care is placebo, because if you recall, Kxalate was grandfathered in. It never went through the modern FDA process of receiving an indication for hyperkalemia. So the first thing you'll see here is yellow and orange. Yellow represents patients with moderate to severe hyperkalemia, and mild represents those patients with mild hyperkalemia. That's in orange. And when you start the patiromer in the green box, you see that the potassium level drops in both patient populations, which is consistent with the phase 1 and phase 2 study. So no surprise there. And then when you transition from the green box to the gray box, you're now going to a different part of the study in which you have instituted placebo control. So you now randomize the patients, all the patients who have just received patiromer and had their potassium level lowered. You split them into two groups. Some of those patients will continue with patiromer as they have thus far, the same dose and the same frequency. And the other group will stop patiromer altogether and be placed in the placebo arm. So now the yellow dotted line represents those who have placebo, and the orange solid line represents those in the patiromer group. And you can see that the potassium level increases almost right away once you stop the medication in the placebo arm. So this was some of the information that the FDA wanted to see to know this medication had an effect that the placebo did not have, and when this medication was discontinued, you reverted back to a more placebo-looking outcome. So the next two slides show the data that was actually published in the scientific literature. So this is the data that the physician community saw. You'll see purple representing those with moderate to severe hyperkalemia, green representing those with mild hyperkalemia, and the yellow is the overall patient population. The baseline starts all the way to the left. The y-axis is your potassium level in the blood. The x-axis is the study duration measured in weeks. Once you start patiromer, at the first time point that you measure the potassium level, which is the third day of the study, you'll note that all the potassium levels drop no matter what your baseline was. And that drop continues as you progress through the weeks of the study, such that you enter and remain within the normal range of potassium between 4 and 5.0. This slide shows what happens when you use patiromer. Compare that group to those who use placebo. So once all the patients have achieved that normal potassium level with patiromer, we took them off patiromer. You saw that 2 slides ago. What exactly happened to the potassium level? So in the placebo arm, you see a rise in their potassium level, a positive increase, which is what we expected. But in the patiromer arm, you don't see any change from that baseline level. And this is part of the information that we provide to the physician community and the FDA to show that there is efficacy of the medication when compared to an appropriate comparator, in this case, placebo. So now we get to the FDA review, in which we provide all of this scientific information to the FDA. But we don't provide all of this information as a bolus. So we are in constant communication with the FDA from the start of our development process all the way to the end, and then even beyond once the medicine is in the marketplace and it is being used by patients. And so the FDA lines of communication are always open all throughout the journey. That way we know what the FDA is looking for. We can make changes to our study designs as indicated and required by the FDA, and we make sure that we're addressing all of their questions so that they get the information they need to make a decision about whether this medicine should be available to the US patient population. We started these communications back in the aughts, and in October of 2015, the FDA approved patiromer for the use of hyperkalemia. And now come the post-marketing efforts. And so if you recall, this is primarily where I operate within the company. The post-marketing efforts are usually what we call life cycle management. So we're going to be managing the life cycle of this medication as it is used throughout the world, in this case, throughout the United States, by patients who have hyperkalemia. So within the life cycle management is education. And so we partner with a number of physician groups and patient advocacy groups like the AAKP to educate the physician community and the patient population about how this medicine should be used, what are its proper indications, what kind of effects can you expect, and what kind of adverse effects can often happen, and if there are any treatments for that, then what those are. So we want to make sure that everyone understands where this medication is appropriately used, how it can be used, and for which patient population it would best serve. Part of lifecycle management also includes changing the label over time. And so the label is really the FDA's way of signaling to the entire community, both physicians and patients, that this medicine has undergone rigorous study and it is approved for particular use. So in the case of patiromer, for example, our FDA label journey started in 2015 when we received approval for the use of hyperkalemia, but there were some warnings. As you can see, there was a warning that this medicine, patiromer, had to be separated from any other medicine at least 6 hours. Of course, that's a bit of a burden on patients to have to space their medicines out 6 hours in either direction if they're taking patiromer. So we performed additional studies, and a year later, that warning was removed and modified that you would have to space the medicine out for 3 hours, but only if you're taking a specific set of other medications. So it's not all medications. And those other medications that need to be spaced out are listed in the label as well. And in 2017, we updated the label again by doing more studies to show that patiromer can be taken either on an empty stomach or with food, that its efficacy is unchanged, depending on whether you choose to eat or not. Then in 2022, we had another label update so that patients could take patiromer mixed in a variety of normal foodstuffs, like beverages, like water, and some soft foods like apple juice. And then most recently, last calendar year, we had a label update change to show that patiromer can be used safely in the pediatric population age 12 to 17 years of age. So part of the life cycle is management is education. The other part is to continue to evolve the FDA label to find new indications for your medication or to refine the existing indications so that there's more granularity and clarity in how the medicine can be used. And there are ongoing efforts once you reach the post-market phase. And so the third part of life cycle management is what we call real-world evidence. So it's one thing to have scientific data that comes out of a structured clinical trial in which everything is monitored and everything is recorded. That's not indicative of the real world. And so once you receive FDA approval for your medication, you often start conducting a number of real-world studies to see how the stochastic environment in which all of us receive healthcare impacts the way a medicine is used and its effectiveness. And if there are any changes that need to be made, can these studies uncover what those changes need to be? And one of the benefits of using patiromer and conducting real-world studies is that it's been on the market for over 8 years. It's been prescribed over 1 million times. So that's a really rich, broad, and deep data set on which we can call upon which I should say we can call so that we can conduct further studies and get a better sense of how this medicine is used in everyday practice. So we're here at the conclusion now. This is, again, the 5 buckets in the journey of bringing a medication from its conceptual design to the patient or the consumer. Again, I operate mostly in the post-market efforts, but we have an opportunity to operate in the first bucket, discovery and development, and in the clinical research bucket as well. You can see the value of each of these buckets, hopefully, in this presentation. So I want to thank you again for your time. My name is Tejas Desai, and I'd be happy to take any questions.
Thank you, Dr. Desai, for your incredible presentation. Before we close, I'd like to ask you a couple questions. Over the last 10 years, we've seen a substantial increase in researchers and industry not only incorporating patients early in the research and development process, but doing so in a substantive way, not a check-off-the-box exercise. What caused the paradigm shift? And in your opinion, why is it critical that patients, who are the end user of the products and services being developed, be part of the solution?
No, that's a really good question. Thanks for asking. So I think that question is in 2 parts. The first is, what do I suspect is the cause of having more patient involvement in the journey of a medication and bringing it to market? And then the second part is, why is that valuable? So here's how I see the cause, and it's a good cause. When patients advocate for themselves, or advocacy groups like AAKP advocate on behalf of those patients whose voice may not be as strong or as present in this particular scientific community, in the research community, that effort over years and years has brought the value and the benefit to the attention of the decision makers in various areas within the scientific community. And that is a good thing, because there are a number of stakeholders in healthcare, and perhaps the most important stakeholder that should have a voice at the table at the beginning would be patients. It isn't enough for the patients to just have a seat at the table, which again was something that had to be fought for, had to be advocated for by patients themselves, their family members as advocates, and advocacy groups like AAKP, but you also want to have a voice at the table. You don't necessarily have to have the same voice as the scientists and the researchers and the business development individuals. You have to bring your own unique voice, because that adds far more to the discussion, and leads to a better product and a better outcome for all the patients that are involved, including those who may not be seated at the table. So I think the cause is advocacy, and constantly reminding the decision makers that there is a value here. And those decision makers have come to the realization, increasingly so, that, and rightfully so, that there truly is a value in having all stakeholders, including the patients, sitting at the table, and earlier the better. So the value, of course, is that you're developing solutions to problems that afflict your fellow human beings, your fellow Americans. And you want to know as clearly as you can the nature of the burden that these problems bring to the lives of the patients, so that you can design solutions that not only work on paper or in a test tube, but can be incorporated readily and easily into the life of a patient. And that can only happen if the patients and their advocates are at the table telling us how these things unfold. How does this disease afflict a patient or a group of patients? What are the hidden factors that we don't see because we may not be afflicted by the condition ourselves, but are important to know because they'll impact whether a patient can be adherent to a medication, whether a patient would be willing to take a medication for a sustained period of time, whether a combination of solutions need to be implemented versus one solution alone. All of these caveats and features need to be considered. They can only come from one source, from the patient and his or her advocate. And they need to be brought into the discussion as early as possible so that we are on the right track and we stay on the right track.
And finally, I myself am a PhD student. Research is my passion, and I've been a part of a number of initiatives such as the Kidney Precision Medicine Project funded by the National Institutes of Health, as well as standing patient councils for AAKP, where I work directly with companies, helping inform the development and design of their research in a patient-centered way. But for fellow patients who are not already in academics, why is it still important for them to understand the product development lifecycle? And how would you encourage them to get involved in research opportunities?
Yes, okay. Thank you for the question. So if you're a patient who hasn't yet delved into the world of scientific research and the development of solutions for the diseases that afflict us, you, or anyone that you know, but you have an interest in doing so, that would be the first step, is having an interest. Know that there is an opportunity and a place for you to sit at the table. So you have to know that first, that the world has changed, our country has changed, such that there is a place for you, your perspective, and your viewpoint in this entire process. Now, once you know that that is true, that it is no longer a dream or an aspiration, other than the fact that we want more patients involved, that would be the biggest aspiration here. The next step would be to determine how you can get involved. And the easiest and probably the most direct way, probably the most efficient way to get involved is to work with your advocacy group first. They often have a lot of channels that are open and are familiar with the stakeholder needs that different companies have, depending on where in the life cycle, in the journey of drug development, they find themselves in with a particular medication. So that's, I would say, the best place to start. Ask your advocacy group like AAKP or any other advocacy group with which you are a member of or aligned with, to determine what options are available, which companies are looking for the patient voice, which ones need more, and what type of perspective are they looking for, depending on the point in the journey in which they find themselves. But the value of being a part of this, I hope, is evident, but I'll repeat it in case it isn't, or in case there's any question or skepticism. The ultimate goal is to bring a solution to patients, to our fellow humans, fellow Americans. And for that to happen, we need to know what you need. We need to know how it fits into your life, whether you can sustain using this medication for the condition you have, or whether there's another condition that takes higher priority and needs to be addressed first before you can address something else. These are things that are difficult to ascertain by just doing research or reading papers. They're far easier and more accurately understood if advocates and patients speak for themselves or on behalf of a larger group of patients. So I hope you see that benefit. It's something we thirst for because we need it in order to make a product that is useful for everyone, not just a select group or an imaginary group of individuals. And it's something that only you can bring to the table through your efforts at asking to sit at the table and sharing your voice.
Thank you again, Dr. Desai. We want to extend our gratitude for your incredible presentation and for being a champion of the importance of engaging patient insights and lived experiences into the R&D process. I know your passion and expertise has truly resonated with our audiences, and we are grateful for the time and effort you put into joining us and delivering such a valuable presentation that I hope inspires more patients to get involved in informing the development of new drugs and devices to advance care.